Okay, okay, thank you. Well, first of all, I would like to thank the organizer of this INRIA-Brasil meeting on digital health for giving me the opportunity to present what I do, what we do at COMMEDIA Project Team. In fact, I was 3 weeks ago, almost 3 weeks ago in Rio Petropolis. I organized, co-organized with people there, Frederic Valentin in LNCC, a wonderful workshop on numerical method for fluids, and we have a wonderful time there. So I hope that I think this kind of meetings are really necessary, and I hope that we can take the opportunity to do things together. The purpose of this small talk is to give you an overview of what we do at COMMEDIA Project Team. So my name is Miguel Fernandez. I'm researcher at INRIA Paris, and I head COMMEDIA, which stands for Computational Mathematics for Biomedical Applications. And so the idea is to share with you in 15 minutes what we do in the team. So this is just as a slide with the people that work at COMMEDIA. So we are around 7 permanent members, all of them, most of the great majority applied mathematicians. Frederic Noller is biomedical engineering. And we have a dozen of non-permanent people. That means PhD students, engineers, or interns and postdoc. And in a few words, what we do is mathematical modeling for biofluids in 2 particular scenarios, which are cardiovascular flows and respiration flows. And basically, we have 3 research axes that you have here, which are, the first one is multi-physics modeling, because this system, as you will see later, are multi-physics. So we couple the different physics that are involved in these systems. We also have an important axis on basically the data assimilation. What does it mean is basically how we couple, we make models interact with data in order to make them predictable, in fact. And then we have what we call a methodological core, where we basically investigate the mathematical problems that arise in the previous 2 axes. Okay, so basically we focus on the applied mathematical aspects of the model. So mathematical analysis, numerical methods development, the numerical analysis of the methods, data assimilation, and then scientific computing. In particular, we spent an important amount of our work on doing software development, in particular scientific computing. So just a few words about what we do in cardiac modeling, and this is related to what Michel Doja presented before. So we are one of the teams at INRIA working on cardiac modeling. And so what we do, so basically I will present for each research axis more or less the motivations of the things that we are doing now, the research directions, and then people we collaborate with. So basically what we do in cardiac hemodynamics is currently not limited to, but related to 2 main motivations, which are using models, mathematical models, to provide mechanistic information to valve repair, in particular mitral valve repair. So cardiac surgeons basically use imaging, and we now know that this is not sufficient to basically predict the outcome of surgical repair. Okay, and so we need mechanistic information, and this is what we plan to do here. Another interesting topic that we are investigating is hypoplastic left syndrome. So this is a very recent work that we do, in particular in collaboration with Assosystem, which is the case of newborns that have a left heart which doesn't work, and so everything has to be done by the right heart. Okay, and so we do fluid structure interaction modeling for this. You have some examples here. So cardiac modeling, where we simulate the hemodynamics in the ventricles, coupling with the muscle, cardiac valves. Here you have the atria too. So an important point here is how to validate the models, and this is something that we do with people in Lyon, HCL, which was also mentioned before by Michel. And so the idea is to use experimental data to basically challenge the models so that they are really predictable. So this is an example of what we do, in fact, in cardiac hemodynamics, 2 examples. So you can use cardiac hemodynamics models to test, evaluate, or assess imaging modalities. For instance, the PISA method, you can evaluate how the PISA method is able to predict the amount of regurgitation in a mitral valve. And you can also use cardiac hemodynamics models to test medical devices, artificial valves, compare medical devices. You have 2 different devices here for the mitral valve. This is the netting valve, and you see by the particles tracking that this one is able to reproduce the natural hemodynamics, and this is not the case. Okay. This is a slide of what we do in respiration. Okay, so let's say that the main motivation, and this was a little bit related to the past COVID pandemic, is to understand the degradation of respiration in patients in intensive care units. Okay, and there is also a link there is also a motivation related to what we do to how to improve respiration control in patients with sleep apnea. And basically what we do is a hierarchy of mathematical models that go from very specific 0D models on ventilation dynamics and oxygen diffusion. And this is a very tricky problem because you have this Aldan Bohr effect, which basically makes oxygen driven the affinity of hemoglobin to other gases, and that is very important to model this. Okay, and so in particular in the degradation of respiration. The idea is also to couple these 0D models with airflow models of long ventilation to have the complete chain. And something that we started to work now also is the simulation of mucociliary transport. Okay, so how mucus is transported in the airways, and this is a very interesting 3D, 1D. So 3D mucus and airflow, and 1D the ciliary, in fact, active structures that basically clean the lungs. Okay, so another example. Now we move to simulation with data interaction. So I will present here 2 examples of what we do in data simulation. So the first one is how to basically calibrate vascular models from 4D MRI imaging. So this is motivated by basically using vascular models to predict aneurysm rupture. Okay, so this is an example too where imaging is not enough to predict the right moment for surgery in aneurysm, but we need a mechanistic information. And one idea to do this is not to remove imaging, but to use imaging to calibrate vascular models. So 4D MRI is very powerful because you have images and then velocity on all the time series. So you have here an example on the right. So you have the images, and then this is the raw, the noisy velocity data. And the idea is to calibrate a vascular model that we have here at the right. So this is only fixed wall, so just flow model, and we do that with different techniques, continuation, observer, etc., etc. Another example of modeling and simulation interaction with data, and this is this one related to how to, I know you all know that you have in your smartphone a photodiode and a LED, so you can basically measure absorption of light in your finger. Okay, this is an example of another device which is not a smartphone but is a thermometer. So you have a LED and a photodiode here, and you can use that to measure light absorption in your tissue. This is used in clinical use to measure oxygen saturation, and the idea is to use this stuff to retrieve blood pressure, okay, to avoid using a tensiometer to monitor blood pressure. And we do that with a company within that developed this kind of devices, okay. And so basically we use poroelastic models of tissue perfusion and light diffusion to simulate that signal. You have the blood perfusion here on the left, and here the absorbed light, okay. And you see the signal. This is what we measure. This is what we simulate, and the idea is to retrieve blood pressure, which is here prescribed from this signal. Another example of simulation data iteration, we have 3 examples. Step 2 is safety pharmacology. So basically is to understand, to predict the impact on drugs from on the cardiovascular signal, system from imaging, from biological sickness. So we did a lot of work on this stuff on cardiac electrophysiology, so impact of on cardiac electrophysiology on drugs, so how this impact the excitation and signal propagation in the heart. And so we start a new approach, which is using the impact on drugs on cardiac tissue, on vascular tissue. And this is an example of what we do. We do that with people in Antwerp. So the idea is to simulate. So you have a piece of tissue, vascular tissue. You put a drug, you excitate the tissue, and the idea is to see the impact of the drug on the tissue. And you see that the model, you have some examples here. So this is the standard without any drug. You put a vasoconstrictor and then a vasodilator, and you see that the model is able to reproduce the experiment, which is the one that you see here simulated. Well, a few words about the methodological core. So this is more mathematical aspect that we look here. So for instance, the question of existence of solutions and well-posedness of the mathematical models that we do. A problem that we look very much now is the mechanical consistency of models for contact in cardiac valves. So this is a very interesting problem because if you basically a standard model do not predict contact. So you see that basically you have here an example. If you have a ball which is basically inside a glass of water, your mathematical model is going to tell you that it never touched the bottom of it, which we know is not true. Okay, and this is because there is a singularity in the pressure that you have here in red, and the idea is to you have this singularity because you don't model roughness, in fact, at the bottom. Okay, and so we need, if you introduce some model of roughness in the bottom, you see that you can remove the singularity. You have something that flatters. Okay, this is an example of the things that we do. We do a lot of work on numerical methods for multiphasic problems, so which go for basically the simulation of cardiac valves. So you have here an example of a very complex multiphasic problem related to respiration and mucociliary transport. So this basically simulate the cilia forest in the lung, and then you have the airflow around it. Okay, and this is a very complex because you have 1D structures interacting with flow, contact, etc., etc. Another important activity in the team is high-dimensional problems. So these high-dimensional problems arise, for instance, in inverse problem in data simulation. So a way of tackling inverse problem in parameter estimation is to make the solution have the parameters of the model as an argument, and then you end up with a problem which has a high dimension. And we use different approaches to solve this. Parametric solver based on tertium approximations, but beyond that, in fact. And we have here an example of we estimate, in fact, the parameters. I mean, this wall motion from velocity measures, so related to 4D flow MRI. And you see that what is nice is that you can, in fact, have some notion of observability, because you see that you can estimate the Lamé parameter lambda 1, but not lambda 2, because you have much more uncertainty. Okay. And then my last slide is about software development. So this is as I mentioned at the beginning, a key cornerstone in the team. So we develop a scientific computer library, finite elements in C++, which basically gathers all what the team or the team members did in the past 15 years. Okay, so this is, we hopefully have an engineer, a permanent engineer from INRIA helping us to maintain this code. So we have fluid mechanics, solid mechanics, electrophysiology, fluid structure interaction, poroelasticity. Okay, and we also, Damiano, one of my colleagues, released a library on tensor methods, so high dimensional problems. So Felice is the vast majority open source. So this is something that we want to share and will be fully open source in the forthcoming months. Okay, so I think I'm done. So thank you again for organizing this.